Basics of Medical Biochemistry. First edition, 2023. Chapter 18. Transcription and Translation. Part 2 of 2. By Professor Dr. Muhammad Jawad. MBBS, Tau, M. Philosophy, Ph.D., Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College Air University, PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. Chapter 18, Part 2 of 2. 18.3 Genetic Codes. The letters A, G, U, and C correspond to the nucleotides containing nitrogenous bases adenine, guanine, uracil, and cytosine respectively in mRNA. They are organized into three-letter code words called codons, and the collection of these codons makes up the genetic code. 20 different amino acids are required for the synthesis of proteins. Since there are only 4 different nucleotides in mRNA, then, each codon must consist of more than a single nucleotide. Codons of 3 nucleotides could provide 64, 43, specific codons. 61 codons code for amino acids and the 3 other codons do not code for specific amino acids. These have been termed nonsense codons that are utilized in the cell as termination signals, stop codons. Stop codons are UAA, UGA and UAG. The start codon of the gene is OG, which codes methionine. Table 18.2 Genetic Codes. Name of amino acid. Symbol of amino acid. Genetic codes in mRNA. Alanine. ALA, A. GCU, GCC, GCA, GCG. Arginine. ARG, R. CGU, CGC, CGA, CGG, AGA, AG. Asparagine. ASN, N. AAU, AAC. Aspartate, aspartic acid. ASP, D. GAC. Cysteine. Cis, C. UGU, UGC. Glycine. GLY, G. GGU, GGC, GGA, GGG. Glutamate, glutamic acid. GLU, E. GAA, GAG. Glutamine. GLN, Q. CAA, CAG. Histidine. His, H. CAU, CAC. Isoleucine. IL, I. AUU, AUC, AUA. Leucine. LEU, L. UUA, UUG, QCUC, Qua CUG. Lysine. Lys, K. AAAAG. Methionine. Met, M. OG. Phenylalanine. PHE, F. UUUUUC. Proline. Pro, P. CCU, CCC, CCA, CCG. Serine. Ser, S. UCU, UCC, UCA, UCG, AGU, AGC. Threonine. THR, T. ACU, ACC, ACA, AC. Tyrosine. TIR, Y. UAU, UAC. Tryptophan. TRP, W. UG. Valine. Val, V. GUU, GUC, GUA, GUG. Stop codons. UAA, UAG, UGA. 18.4 Mutation. The mutation is a stable change in the DNA structure of a gene, which may be expressed as a phenotypic change in the organism. Mutations may be classified into the following major types. 1. Base substitutions. It may be a transition, in which one purine pyrimidine base pair is substituted by another. Example: AT is substituted by GC. Figure 18.13 Transition type of base substitution, at GC. In the case of the transversion type of base substitution, one purine pyrimidine pair is transversed. Example CG is transversed by GC. Figure 18.14 Transversion type of base substitution, CG-GC. 2 frame shift mutations, deletion or insertion of base pair results in a change of reading frame. Figure 18.15 Frame shift mutation. 3-point mutation. Isilent mutation, base pair substitution results in the same amino acid. They may occur in a region that does not code for a protein, or they may occur within a codon in a manner that does not alter the final amino acid sequence. The protein function is not altered. 2 nonsense mutation, base pair substitution results in a stop codon that causes the formation of a shorter polypeptide, i.e., truncated protein. 3 missense mutation, base pair substitution results in the substitution of a different amino acid. It may be conservative when a substituted amino acid is of the same class or non-conservative when a substituted amino acid belongs to a different class. Table 18.3 Sites and types of point mutation. Site of change. No mutation. Point mutation. Silent. Nonsense. Missense, conservative. Missense. Non-conservative. DNA level. TTC. TTT. ATC. TCC. TGC. mRNA level. AAG. AAA. UAG. AG. AC. Protein level. LIS. LIS. Stop. ARG. THR. 18.5 Translation. The translation is the process of synthesis of protein and polypeptide chains, having a specific sequence of amino acids with the help of mRNA and tRNA in the ribosomes. A complete set of proteins expressed in a cell is called a proteome. The cell must possess the machinery necessary to translate information accurately and efficiently from the nucleotide sequence of an mRNA into the sequence of amino acids of the corresponding specific protein. The process of translation is quite complex but very rapid, as a cell can produce a polypeptide of 1,500 amino acid residues just in 5 seconds. Prokaryotic mRNA is polycistronic as it has many coding regions, whereas eukaryotic mRNA is monocistronic as it has only one coding region. 18.5.1 Translation requirements. Genetic codes are required for translation, during which nucleotide sequence on mRNA is translated to the specific amino acid sequence. Following are the essential requirements for the normal process of protein synthesis, i.e., translation. 1. Availability of all the 20 standard amino acids. 2. mRNA, template. 3. Functionally active ribosomes. 4. Amino acid activating enzymes, aminoacyl synthetase. 5. tRNAs, at least one specific for each amino acid. 6. Protein factors, initiation, elongation, and terminating factors, IFS, EFS, and TFS. 7. Peptidyltransferase, which catalyzes peptide bond formation. 8. ADP and GTP as sources of energy. Role of tRNA in protein synthesis. The role of tRNA in protein biosynthesis is to carry amino acids to the site of protein synthesis in ribosomes and to align them together according to the sequence of the codons in the mRNA. 3 bases in tRNA that can pair with the codons in mRNA are known as anticodons. For example, UUU on tRNA is complementary to AAA on mRNA. Each tRNA carries its corresponding amino acid to the 3' end, which has a sequence of CCA. A set of enzymes called amino acyltransferase synthetases are required to charge the tRNA with the proper amino acid. During protein synthesis, anticodons form temporary hydrogen bonds with the codons. The direction of mRNA reading is 5 to 3. The codons run in the 5 to 3 and the anticodons in the 3 to 5 directions. Thus, the codon for methionine is 5-OG3 and its anticodon is 3-UAC5. Role of ribosomes in protein synthesis. Ribosomes are known as the translational apparatus of the cells. They are found free in the cytoplasm or very close to the endoplasmic reticulum. They are about 20 nanometers in diameter and are composed of 60% rRNA and 40% proteins. Eukaryotic ribosomes generally have a Svedberg S value of the 80s and are comprised of small 40s and large 60s subunits. The S value is not strictly additive. There are 3 adjacent tRNA binding sites on the ribosome. IA site, aminoacyltRNA binding site. 2P site, peptidyl binding site, for the central tRNA molecule containing the growing peptide chain. 3E, exit, site is occupied by the empty tRNA as it is about to exit the ribosome. Figure 18.16e, exit, P, peptidyl, A, amino acid site on the ribosome. In addition to the APE sites, there is an mRNA binding groove that holds onto the message being translated. 18.5.2 Steps of translation. Following 4 distinct steps are involved in the process of translation. 1. Activation of amino acids. 2. Initiation. 3. Elongation. 4. Termination. 1. Activation of amino acids. Formation of aminoacyltRNA complex. Aminoacylation is the process of adding an aminoacyl group to a molecule of tRNA. It produces tRNA molecules with their CCA3 ends covalently linked to an amino acid, charged tRNA. This step takes place in the cytosol. It requires 20 amino acids, 20 tRNAs, 20 enzymes, aminoacyltRNA synthetase, ADP, and Mg2+. Each of the 20 amino acids is covalently esterified to their specific tRNA under the influence of their corresponding aminoacyltRNA synthetase enzyme. AminoacyltRNA synthetase can perform proofreading, editing, as it can remove incorrect amino acids if get attached to tRNA. ADP AMP plus PP. Mg2 plus. Amino acid plus tRNA aminoacyltRNA. The initial amino acid in prokaryotes as well as polypeptides synthesized in mitochondria is N-formylmethionine only. The linkage takes place at carbon number 3 of the ribose part of 3-terminal adenosine of tRNA. There are 2 levels of control to ensure that the proper amino acid is incorporated into protein. The first is charging the proper tRNA and the second is matching the equivalent tRNA to the messenger RNA. Figure 18.17 The mechanism of aminoacylation of tRNA. 2. Initiation of polypeptide chain formation. The initiation of polypeptide chain synthesis needs I initiation factors, If1, If2, and If3 in prokaryotes and many initiation factors in eukaryotes symbolized as EIF. 2. Initiating aminoacyltRNA. 3. Initiation codon and mRNA. A Schindel-Garneau sequence, in E. coli, 5 UAGAG 3, situated 6 to 10 N bases upstream of OG codon. B. OG sequence in human beings. 4. Ribosomal subunit. VGTP and Mg2+. The initiation is the assembly of the translation machinery before peptide bond formation. Protein synthesis is initiated at the 5 terminals i.e., cap end of mRNA. It occurs in the following 4 stages. I. Complete separation of the whole ribosome, the ADS, into 60s and 40s subunits by EIF6. 2. The start codon OG is identified by a special initiator tRNA, tRNAi. 40s subunit, 30s in prokaryotes, binds with If2 and metRNA, at the expense of GTP to form 43s subunit. In bacteria and mitochondria, N-formylated methionine, Fmet, is carried by tRNAi. 3. If4 facilitates the binding of mRNA with 43s to form 48s. This complex scan mRNA for OG. 6. 60s subunit binds to 48s to form an initiation complex. At this point, metRNA is on the P site ready for elongation, the A site is free and ready to accept the next charged tRNA. The difference in the initiation of prokaryotes and eukaryotes. Initiation is controlled differently in prokaryotic and eukaryotic ribosomes. In prokaryotes, specific sequences in the mRNA around the OG codon, called Schindel-Garneau sequences, are recognized by an initiation complex consisting of a met-aminoacyl tRNA, initiation factors, IFs, and the small subunit of the ribosome. In contrast, eukaryotes use a scanning mechanism to initiate translation. Eukaryotic mRNAs have a distinct structure at the 5 ends. 3. Elongation of polypeptide chain. The direction of polypeptide chain synthesis is from NH2 terminal to COOH end. The addition of each amino acid takes place at the carboxyl end of the growing chain. The elongation process needs I initiation complex. 2. Next amino acyltRNA. 3. Elongation factors, F2 GTP, FTS, and GTP in prokaryote while F1 GTP and F1 gamma in eukaryotes. 4. Enzyme peptidyltransferase. The elongation of the polypeptide chain is a cyclic process. It occurs in the following 3 stages. I. New charged tRNA binds to mRNA on the A site by complementary base pairing. Incorporation of the correct amino acyl tRNA is determined by base pairing interactions between the anticodon of the tRNA and the codon of a messenger RNA. A proper reading of the anticodon is an important quality control step ensuring accurate protein synthesis. 2. The ribosome catalyzes the transfer of the amino acid from the P site onto the amino acid at the A site, forming a new peptide bond. This stage is catalyzed by a peptidyltransferase. 3. The ribosome moves along the mRNA to the next codon towards the 3 ends. This movement requires an elongation factor called translocase and energy in the form of GTP. The empty tRNA at the P site is displaced into the E site, and the tRNA that has the growing peptide chain on it is moved from the A site to the P site. There is a tunnel through the large subunit that allows the growing polypeptide chain to pass out of the ribosome. The process is then repeated. Transpeptidation, the transfer of peptide on tRNA at the P site to the amino acid on the tRNA at the A site is known as transpeptidation. Translocation, after transpeptidation, the movement of mRNA-3 nucleotides toward the 3 end is known as translocation. The process of translocation needs energy from the hydrolysis of GTP and FG-GTP in prokaryotes while F2-GTP in eukaryotes. Polysome, polyribosome. The complex of one mRNA and many ribosomes is called a polysome. It is formed because the length of most mRNA is such that it requires more than one ribosome for translation. 4. Termination of polypeptide chain formation. This step requires ADP, termination codon on mRNA and polypeptide release factors. The termination of protein synthesis is signaled by one of the 3 termination codons on mRNA via UAA, UGA or UAG, which do not code for any amino acid. These codons have no corresponding tRNA that can pair with them. Stop codon causes a release factor to bind this open site, causing the peptide to be released from the 3 ends of tRNA at the P site. After the release of the polypeptide chain, the ribosome and mRNA also dissociate. Ribosomes after releasing polypeptides become free and restart another cycle of protein synthesis. Release factors, RF in prokaryotes. Example in E. coli. The release factors found are RF1, RF2, and RF3 GTP. RF1 can recognize stop codons UAA and UAG, whereas RF2 can recognize UGA and UAA. RF3 GTP causes the release of RF1 or RF2 as a result of the hydrolysis of GTP. In eukaryotes, all 3 stop codons can be recognized by a single release factor, i.e., ERF. There is another release factor in eukaryotes as ERF3, it worked like RF3 of prokaryotes. 18.5.3 Regulation of translation. The expression of most genes is controlled at the level of their transcription. Transcription factors, proteins, bind to promoters and enhancers, turning on, or off, the genes they control. However, gene expression can also be controlled at the level of translation. One method in eukaryotes is the covalent modification of EIF2 via phosphorylation to make it inactive. There are inhibitory proteins both in prokaryotes and eukaryotes that bind to mRNA and block the process of translation. Figure 18.18 Steps of protein synthesis. 18.5.4 Post-translational modification. Newly synthesized proteins are immature or nascent proteins that do not attain their final biologically active structure until they have been altered by one or more processing reactions called a post-translational modification. Post-translational modification of proteins is detected by mass spectrometry or Eastern blotting. Following are the common methods of a post-translational modification. 1. Trimming. Many proteins are formed as large precursor molecules that are functionally inactive and part of their chains must be removed to release the active molecules. For example, proenzymes, zymogens, such as trypsinogen of trypsin and chymotrypsinogen of chymotrypsin respectively. Most nascent proteins have methionine as the first amino acid because the start codon on mRNA also codes for this amino acid. Methionine is usually removed during post-translational modification. 2. Covalent modification. It occurs by the following reactions. I-phosphorylation, addition of phosphate to hydroxyl groups of serine, threonine, or tyrosine residues of proteins. It takes place by protein kinase and is reversed by phosphatase. The phosphorylation may cause an increase or decrease in the functional activity of the concerned protein. 2. Hydroxylation. Proline and lysine of procollagen are hydroxylated to give hydroxyproline and hydroxylysine present in collagen. 3. Carboxylation. Carboxylation of glutamic acid residues in prothrombin to form carboxyglutamate. 4. Glycosylation. It is the addition of carbohydrate moiety to the synthesized protein. Carbohydrate moiety, which is usually an oligosaccharide, is attached to an asparaginase by N-glycosidic linkage or to serine and threonine by O-glycosidic linkage. V. Disulfide bond formation. The disulfide, SS, bonds are formed between neighboring cysteine amino acids as intra- and interchain disulfide bridges. For example, in insulin molecules. The first interchain disulfide link is found between cysteine residue found at position 7 of both chains, while the second interchain disulfide link is found between cysteine residue present at position 20 of a chain and 19 of B chain. The insulin molecule also contains an intrachain disulfide bridge between two cysteine amino acid residues found at positions 6 and 11 of the A chain. 18.6 Mitochondrial protein synthesis. Only a small number of proteins are synthesized by mitochondria. Mitochondria contain their DNA and protein synthesizing machinery. Mitochondrial genetic codes are slightly different. Codon-anticodon interactions are simple and managed only by 22 species of tRNA. Most mitochondrial proteins are coded in the nucleus and transported into mitochondria. Table 18.4 Antibiotics as inhibitors of protein synthesis. Antibiotic. Mechanism of action. Streptomycin, including other aminoglycosides. Inhibit initiation and also cause misreading of codons. Tetracyclines. Bind to the 30s subunit of the ribosome and block its binding to amino acyltransferase. Chloramphenicol. Inhibits the activity of peptidyltransferase. Erythromycin. Binds to 50s subunit of ribosome in prokaryotes and inhibits translocation. Purimycin. Causes premature termination of polypeptide chain synthesis. Cyclohexamide. Inhibits the activity of peptidyltransferase 60s subunit of ribosome in eukaryotes. Diphtheria toxin. Inhibits elongation factors. 18.7 Chaperones. Some proteins are not folded spontaneously as they are synthesized in the cell. Their proper folding is facilitated by the action of specialized proteins called chaperones. One major function of chaperones is to prevent both newly synthesized polypeptide chains and assembled subunits from aggregating into non-functional structures. The folding process in some proteins starts at N-terminus as soon as it arises from the ribosome. This results in the formation of a molten globule. The formation of the molten state is complete within a few seconds. It is then followed by comparatively a slow process of reorganization of side chains to get the correct tertiary structure, which takes place in several minutes. Misfolded proteins are a burden over a cell in two aspects. One is a significant loss of energy and the other is abnormal clumping, and their aggregates can cause many serious diseases. Chaperones are effective tools to correct misfolded proteins. They bind to the exposed hydrophobic patches on the misfolded proteins and perform mechanical work to achieve accurate configuration. The required energy is obtained by the hydrolysis of ADP. The proteins corrected by chaperones are called client proteins. The two common types of chaperones are HSP60 and HSP70. HSP equals heat shock proteins. They are produced in the condition of elevated temperature, which is the result of the production of large amounts of misfolded proteins in the cell. HSP70 comes into action early in the correction process. It combines with the polypeptide chain emerging from ribosomes at its 7 hydrophobic amino acids. The function of HSP70 is supported by HSP40 and also aided by the hydrolysis of many ATPs to fold a single polypeptide chain correctly. Fully synthesized proteins are corrected by HSP60. It is also known as a chaperonin. It forms a large barrel to facilitate easy isolation of proteins and also provides a better environment for correct folding. It holds the proteins at the rim of one of its barrel, then the binding of ADP increases the diameter of the barrel to provide sufficient space to partly unfolds the protein. Inside the barrel, protein can be easily folded. After that, binding and subsequent hydrolysis of ADP causes the weakening of association. The properly folded protein is released on the binding of another ADP. If folding is not completed, then the cycle is repeated. Clinical aspects: a soluble protein that is normally secreted from the cell in a misfolded state is converted into an insoluble extracellular amyloid fiber. The aggregates of these insoluble extracellular amyloid fiber deposits in various sites produce a condition called amyloidosis. There are many human genetic disorders like diabetes mellitus type 2, Alzheimer's disease, Huntington's disease and Parkinson's disease are associated with misfolding proteins. Figure 18.19 Role of chaperone and chaperonin in protein folding. Summary. The flow of information in the cell starts with DNA, which replicates to form more DNA. Information is then transcribed into RNA, and then it is translated into protein. Transcription is the process of copying genetic information contained in DNA in the form of RNA. The translation is the process of synthesis of protein and polypeptide chains, having a specific sequence of amino acids with the help of mRNA and tRNA in the ribosomes. In eukaryotes, the processes of transcription and translation are distinct both spatially and in time. In prokaryotes, which have no nucleus, both transcription and translation occur simultaneously. Prokaryotic RNA polymerase is a zinc-containing multi-enzyme system that can recognize the promoter region at the beginning of the length of DNA that is to be transcribed. The promoter region is the nucleotide sequence at the beginning of a stretch of DNA that is to be transcribed. The process of synthesis of RNA in prokaryotes involves the binding of RNA polymerase to the DNA double helix, the unwinding of the DNA helix, initiation of RNA synthesis, elongation of the RNA chain and termination of RNA synthesis. The initiation starts with the attachment of RNA polymerase to the -35 sequence of the promoter region. Sense or coding, non-template, strand is the strand of DNA that is used for coding. It has the same sequence of N bases as the RNA transcribed, except for thymine and sugar. Antisense or non-coding, template, strand is the RNA-forming strand of DNA. It is a non-coding strand. Elongation of the RNA chain occurs in the direction of 5 to 3 antiparallel to the DNA template. Termination of RNA synthesis is signaled by a sequence in the template strand of the DNA molecule. Rifampicin or rifampin, an antituberculosis drug, can inhibit the initiation. There are 3 types of RNA polymerase in eukaryotes: RNA polymerase I, responsible for rRNA synthesis, RNA polymerase II, which synthesizes the mRNA, and RNA polymerase III, which synthesizes the tRNAs and 5's rRNA. After the synthesis of a short stretch of mRNA, RNA polymerase II physically moves forward and leaves the promoter region. This is called promoter clearance. Elongation of the mRNA chain is the addition of nucleotides to the three ends of RNA stretch formed on the DNA template during initiation. Termination of mRNA synthesis in eukaryotic transcription is sensed by two types of particular sequences on the newly synthesized RNA which act as termination signals. These are AAUAA and CA. Primary tRNA and rRNA of both prokaryotes and eukaryotes are post-transcriptionally modified by ribonuclease. Post-transcriptional modification in mRNA, in eukaryotes only, includes 5-capping, the addition of poly-A tail and the removal of introns. The letters A, G, U, and C correspond to the nucleotides containing nitrogenous bases adenine, guanine, uracil, and cytosine respectively in mRNA. A mutation is a stable change in the DNA structure of a gene, which may be expressed as a phenotypic change in the organism. During protein synthesis, anticodons form temporary hydrogen bonds with the codons. The direction of mRNA reading is 5:3. Ribosomes are known as the translational apparatus of the cells. The steps of translation are activation of amino acids, initiation, elongation, and termination. Activation of amino acid is the formation of amino acylternate complex. The initiation is the assembly of the translation machinery before peptide bond formation. The direction of polypeptide chain synthesis is from the NH2 terminal to the COOH end. Termination of polypeptide chain formation requires ADP, termination codon on mRNA and polypeptide release factors. The post-translational modification includes trimming and covalent modifications like phosphorylation, hydroxylation, carboxylation, glycosylation, and disulfide bond formation. Antibiotics such as streptomycin, tetracycline, and chloramphenicol can act as inhibitors of protein synthesis. References. Exercise questions. For MCQ 1-5 see Part 1 of this chapter. MCQ 6. Which of the following is the specific function of single-strand binding protein, SSBP, during the replication of a DNA molecule? Formation of Okazaki fragment. Formation of replication fork. Identification of the origin of replication. Keep two strands of DNA separated. Resolution of supercoils. MCQ 7. Which of the following is the main distinguishing feature of reverse transcriptase, RNA-dependent DNA polymerase, in comparison to DNA polymerase? Contains 3 to 5 exonuclease activity. Follows Watson-Crick base pair rules. Synthesizes DNA in the 3 to 5 direction. Synthesizes DNA in the 5 to 3 direction. Can insert inosine into a growing DNA chain. MCQ 8. Which of the following characteristic events occur in patients with xeroderma pigmentosum? Inability to exercise a section of the UV-damaged DNA. Inability to synthesize DNA across the damaged region. Loss of proofreading. Mutation in the primase gene. Mutation of one of the mismatch repair components. MCQ 9. In addition to an RNA that is complementary to the telomeric repeat sequence, which of the following enzymes is also required for the normal activity of telomerase? DNA-dependent DNA polymerase. DNA-dependent RNA polymerase. DNA helicase. RNA-dependent DNA polymerase. RNA-dependent RNA polymerase. MCQ 10. A 6-year-old girl presented with severe skin blistering on her arms and back after sun exposure for 15 minutes without sunscreen. Numerous telangiectasias were also observed. Xeroderma pigmentosum was suspected in this girl. Which of the following processes is most likely to be defective in this patient? DNA polymerase proofreading activity. Double-strand break repair during replication. Photoactivatable thymine dimer removal. Removal of uracil from DNA. Single-strand break repair during replication. End of Part 2 of 2 of Chapter 18. Thank you.